Thank you. All right, the fear of holes. It's something that a few of you may have heard of before. Basically, it's the fear specifically of clusters of holes. Something that got popular about the early 2000s, was spread around quite a bit on emails and GeoCities, if anybody remembers GeoCities. We're talking some old school internet stuff here. Gained a lot of popularity when it was added onto trypophobia, or excuse me, trypophobia was added on urbandictionary.com as the intense and irrational fear of holes. Well, that spurred the interest of the people at Popular Science, who investigated a little bit further and discovered, well, trypophobia is not an actual phobia. It doesn't show up in the DSM, but there are some pretty interesting things about it. One thing for sure is there are a lot of people who claim to have trypophobia. In fact, many websites have been dedicated to trypophobia and the fear of holes, including trypophobia.org, trypophobia.com, trypophobia.net. We have the whole variety of .nets up here, including a Facebook group that has over 4,000 users. So there's definitely something going on, but until recently, it hasn't been very much sciencey. What we do know about phobias is that a specific phobia is a marked persistent fear that is excessive or unreasonable, cued by the presence or anticipation of a specific object or situation. So in this case, it would be an object with holes in it, or a situation in which you're present around something with a ton of holes. What we do know is that an aversion does not quite meet these criteria to be considered a phobia. Exposure to a phobic stimulus almost invariably provokes an immediate anxiety response. So we'll talk a little bit further about this distinction between phobia and aversion. But that did introduce it to the scientific realm, in which Cole and Wilkins presented some research that they conducted on trypophobia. They started out by asking adults at the University of Essex, whether or not they liked this photograph. Of the 286 adults that they questioned, 11% of males and 18% of females reported that this image was either uncomfortable or even repulsive to look at. So there's obviously a large set of individuals that don't quite like this type of imagery. So to further investigate, they invited undergraduate students to view images that they had gathered from trypophobia.com to investigate if they see these photos differently than they see other photos of regular holes, things like pipes, straws, hula hoop, things like that. What they did find is that trypophobic images are different. And the way they went about that is by analyzing the spectral analysis of the photos. This includes two main domains, which is the luminance contrast and the spatial frequency. These two components of every image gathered together to bring to us what the actual image is representing. In this image here, I have a low contrast versus high contrast image, a low spatial frequency versus high spatial frequency image, and what the image looks like when combined. So when examining these components of trypophobic images, Cole and Wilkins found that trypophobic images differed significantly from images that appear in nature. In fact, images that appear in nature typically have a contrast increase, excuse me, the contrast increases as the spatial frequency decreases. There's a pretty common rule of thumb for images that come from nature. Trypophobia strays from this standard. In fact, not only did it significantly stray, there was more aspects that were represented in the study that they did that showed they were quite different from normal images of nature. Here I have pictured a blue ring octopus, a cone snail, and a blue spotted tree frog, all of which are poisonous, versus their non-poisonous or non-dangerous analogs. Cole and Wilkins found that the animals that were poisonous matched the same specifications as trypophobic imagery. That high mid-range spectral frequency is a common characteristic in not only trypophobic images, but of animals that are considered dangerous. So looking further into the physiological and psychological aspect of trypophobia, we conducted an experiment that examines the physiology behind trypophobia via electrodermal activity, which I will talk about. We evaluate a trypophobia scale that was produced by Ann Lee, who is one of the colleagues of Cole and Wilkins. And we looked for other possible influences that may be involved in this supposed trypophobia. In general, we asked, do general anxiety levels play a role in trypophobia? Can a scale successfully measure and predict trypophobic responses? And do reported comfort levels match physiological reactions? The way we designed this experiment was we brought in 35 undergraduate students who were asked to participate in an experiment on fear reactions. Participants were given the a generalized anxiety disorder scale, which was a 7-item scale, a pretty quick analysis of what the participants' general anxiety levels are. We looked at the trypophobia scale that Ann Lee provided to assess their level of trypophobia. And before presenting trypophobic trigger images and control images, we attached these students to an electrodermal activity device. So over here I have a small depiction of how the images were rated. They're rated from -5, which is least comfortable, 0 indifferent, or a 5 for the most comfortable. This picture of a puppy would probably get a 5. At least that's what we would expect. So a little bit more on electrodermal activity. The way electrodermal activity works is by sending a small amount of electricity through 2 points on the skin. As somebody becomes psychologically aroused, the pores in your skin open up, allowing the salinity in the skin to conduct more electricity than when you are calm or in a calm state. This electrical flow is measured in microsiemens, which gives us a really strong indicator of what a person's psychological status is at the time that we measure. Our basic layout was we started out with a 60-second slide of a nice, calm prairie. This allowed the participant to reach baseline levels of electrodermal activation. This allowed us to get things set up on our end as well. We then had 5-second blink slide intervals in between before we began to show the next slide. In this image here, I have an example of a trypophobic trigger image followed by a blink slide, followed by a control image. What we were looking for through the psychophysiological reaction was the level of electrodermal activity. So as you can see from here, we have a baseline, which is common at the beginning of the experiment. We have a stimulus marked by this green dropper. We have a reaction. In this case, we have a trypophobic image. We're expecting to find greater physiological reaction or increase in skin conductance response, followed by a blink slide to allow baseline to be reachieved, followed by the introduction of another stimulus. In this case, a control image, picture of cheese, shouldn't present as much of a trypophobic response as the stuff over there that I'm not exactly quite sure what it is. So by viewing these electrodermal reactions, we're able to plug this information into SPSS and evaluate what exactly is going on when somebody views a trypophobic image versus when they view a control image of a regular hole. Going back to Ann Lee's trypophobia scale, we were able to find that his scale was able to measure and predict trypophobia accurately. That is, higher scores on trypophobic levels, which is indicated by the axis over here. Oh wait, oh, the laser. Technology. This stuff is amazing. Over here, we have, as the trypophobia level increases, the comfort level to trypophobic images decreases. Interestingly, and very importantly, trypophobia level, as it increased, did not predict any change in the comfort level of control images. So we are looking specifically at trypophobic triggers, and not just in general at holes. Out of the 35 students that we evaluated, 6 had trypophobia levels that, according to Ann Lee's trypophobia scale, would be considered trypophobic. Some interesting things came out when we looked specifically at those 6 students. That was mainly that those students did not score any higher on the generalized anxiety disorder scale than the rest of the population. Trypophobic participants also rated trypophobic triggers as much less comfortable than control images compared to the rest of the participants. And the 6 participants did not rate the control images, the images of regular holes, as any less comfortable as the rest of the participants. Overall, we found that there is no relationship between the generalized anxiety disorder level of a participant and their rating of trypophobic images. Going back to the electrodermal activity, we're able to find that our participants reacted at a much greater physiological level when viewing trypophobic images. That is, their skin conductance, which is a representation of their psychophysiological reaction, was intensified when viewing images of trypophobic holes versus images of regular holes. Also, they did not show higher electrodermal activity when viewing control images. This was consistent across both the peaks and the means of the electrodermal activity. So this is something that we're looking at as an overall psychological and physiological reaction that's specific to trypophobic images. So in general, what we found is that trypophobia does not necessarily meet the specific criteria for being a phobia, but there is some spectral qualities of trypophobic images that separate them from typical nature images. This is particularly that high mid-range spatial frequency. Participants who looked at these trypophobic images had higher skin conductance responses, and interestingly also, females reported less comfort with trypophobic images than males. This corroborates with what Colin Wilkins found in their initial studies. So moving forward, aversive reactions, to trypophobic images are seen commonly throughout the general population. This would explain why there's such a large internet base of people sharing pictures of trypophobic triggers. Generally, reactions like this can be considered innate because it's so prevalent throughout the human population, and because these reactions happen to us on a physiological level, it's our place to assume that trypophobia is likely a result of Darwinian aversion foundations similar to fear of snakes, spiders, heights, and other stressors that were represented throughout our evolutionary history. That question, of course, needs to be tested. Like the question of why some spotted animals are dangerous and others are not so much. So it's not exactly as simple as saying that all spotted creatures are dangerous and that's exactly why we fear these holes or high mid-range spatial frequencies. There's some other components at play here that we'll have to investigate further. Also, if those spectral properties of that high mid-range spatial frequencies is eliminated from a trypophobic image, does that alter a person's physiological response? Lastly, how can we apply this and adapt this to a biological model such as Batesian mimicry, and how some animals may use this to their advantage, and how this may have developed through the course of evolution. In conclusion, we can find that scientific questions like, what the heck is trypophobia, can come from almost anywhere, including Reddit, GeoCities, or an email. Sometimes these questions lead to actual science, and which people like Dr. Pipitone can start conducting research to get answers to these questions, but in the end, what we see is if we know more at the end of a study than what we knew at the beginning, we've done something pretty incredible. I want to thank everybody that was involved. Of course, Dr. Pipitone, Haley, Danielle, Will, Eileen, and everybody else who is involved with Student Scholar Days. This is a great opportunity for me to present to you. I'd like to thank you very much. Oh, yes. Yes.
So do you suffer from trypophobia?
You know, that's an interesting question. When I started out with this, I was dead set in saying that no, I don't suffer from trypophobia. Throughout the course of time, looking at these holes repeatedly and almost every day going in and seeing some sometimes quite uncomfortable imagery, I found not only that I see holes everywhere, but I am slightly more averted to them than what I started out with. So can I say I'm a trypophobic? Not quite, but definitely more aware of them now. Sure. Any other questions? Yes?
Can you talk a little bit about the EDA and how that you think added to what you did on top of the scale system, or that kind of thing?
Yes. So what we're looking at with the scale system was mostly trying to corroborate with Ann Lee, who was able to create this scale system as a measure of trypophobia. In a way separate from that, the electrodermal activation that we looked at was more specific towards innate and unconscious reactions to the imagery. We used electrodermal activity not only because it's one of the more robust measures, but it's also pretty common in these types of studies, particularly things that look at fear reactions and cognitive stressors. Electrodermal activity is something that you can't control, so the person that's strapped into the machine, and which of course it's also a component to a polygraph lie detector test, can't control what their body is physiologically reacting to. So when we displayed those images and we saw a jump in skin conductance response, we knew then that we can't say exactly that they were comfortable or uncomfortable. What we can say is that they were then stressed by that image. And seeing that trypophobic images led to an increased electrodermal activity versus the control images, we're able to say that there's something specific about trypophobic images that causes physiological reaction.
Yeah. No, I think that's awesome. It adds to it.
Yeah.
It's like having that physiological component, because you could say, oh, well, they're just responding that way because
Right.
It adds some more to make it stringent. So
Correct. Correct. Any other questions? Yes?
When did we start noticing that there was such a thing as a fear of holes, and how far back in time of man's understanding did we start developing these fears for, and now we're starting to notice here now today?
It's a good question. The fear of holes in specific doesn't have a very long history, at least not a long recorded history. One of the popular types of images that you can find on the trypophobia.com website is photoshopped images of like the lotus seed pod that I had toward the beginning of the slide, photoshopped onto people's skins. Those look like lesions. The aversion towards lesions and the aversion towards things in our environment that may represent something harmful is something that has been well documented. I unfortunately am not as knowledgeable in that field, but that is something that we see as a developed pattern, not only in humans, but in many animals, that the type of imagery that is received by an animal will send more information than just that animal, but along with it, innate mechanisms such as approach or aversion mechanisms. Sure. Yes?
So I obviously must have trypophobia because the minute you put that up there, I have to turn my face, and I happen to look down at this gentleman's shoes on the bottom of the shoes are red and there's holes in there. And so when I just Googled trypophobia and I just did the images and it's that girl with the lesions right away.
Yeah.
I start itching really bad. So what is a cure for? I mean, what can you do?
Well, first of all, I'd like to see you in my lab. You know, right now there's not much that we could say as far as curing trypophobia. As far as what we know and what the study basically sums up is that it is something innate. It's something that's deep down inside of every one of us. When we see these high mid-range spatial frequencies, we freak out. So what I did notice is that if I go online and look at those "Are you trypophobic?" tests, by the end of them, I would say I am 10, at least 20 times more afraid of trypophobic imagery than not. And especially the ones where things are photoshopped, because there's some weird things out there on the internet.
Well, it's funny because I know most of you guys have gone down to Lolita Pass, and there's a certain part of the very end where it clears off. There's a mountain, and if the snow hits it a certain way, in my mind, I can see like the weeds or whatever it is, and I see holes, and I just literally will start scratching my head.
Interesting.
I can see, it looks like a head with dandruff and just holes.
Interesting.
It just freaks me out.
Wow, that is very interesting. Well, like I said, when can we schedule you to come in? You can help us figure out what we need to do. You'll be participant number 1 in treating trypophobia. Well, were there any last questions? Thank you very much.